Good afternoon, everyone. Welcome. I'm Jack Van Horn from the University of Virginia. It's my distinct pleasure to welcome you to this edition of our 2024-2025 Foundations of Biomedical Data Science Seminar Series. Our unique lecture series is made possible through the generous support of the Integrated Translational Health Institute of Virginia, the UVA College of Arts and Sciences, the UVA School of Data Science, and through grants from the National Institutes of General Medical Sciences. Since its founding in 2016, the Foundations of Biomedical Data Science Seminar Series has been at the forefront, exploring the transformative impact of data science methodologies, artificial intelligence, deep learning, and statistical modeling across diverse themes in biomedical and health sciences. Our selected theme for this year is focused on quantitative approaches in spatial multi-omics for guiding personalized medicine, where each Friday we enjoy presentations from a wide range of leading thinkers about the issues, the promise, opportunities, and the hurdles associated with advanced genomic, transcriptomic, metabolomic, and other omic methods, along with a wide variety of data analytic opportunities. We are pleased to announce that due to some of the recent things that have gone on with the things in the recent government, that there is a bright side in that we have extended our 2024-2025 application cycle for our Biomedical Data Science Innovation Lab to February 14th to give people extra time to make their plans to be able to join us in Nashville, Tennessee, where we'll be having our Biomedical Data Science Innovation Lab program at the Hutton Hotel right there in Music City in downtown Nashville in June of 2025. We're very excited about that program where junior faculty, biomedical, and computational researchers come together to develop novel joint projects on topics of quantitative approaches and applications for spatial multi-omic data analysis and use those towards grants and potential literature reviews. So for today's seminar, however, I am delighted to welcome our speaker, Amanda Seyerle from the University of North Carolina. Dr. Seyerle is an assistant professor where her research focuses on molecular etiology of adverse drug reactions and variable drug response. A critical element in her work is the inclusion of diverse populations to expand the generalizability, inference, and impact of precision health advances driven by multi-omics data. In the spirit of our 2024-2025 Biomedical Data Science Seminar Series theme, her lecture today is entitled "The Challenges and Opportunities of Incorporating Diverse Populations in Multi-omics Research." Amanda will describe how multi-omics research has brought about new advances in precision medicine. However, it also has the potential to widen existing health disparities. It is critical to consider the role of diverse populations in multi-omics, and Amanda will give us some guidelines on how to wrangle this when designing studies to advance precision health. As always, we are streaming this lecture live and for recording via YouTube. If you're watching us on YouTube, thank you so much for joining us. Also, our specially selected 2024-2025 Biomedical Data Science Innovation Lab participants, as well as alumni, are encouraged to submit any questions via the chat feature in their YouTube sessions. I'll be synthesizing these questions and asking them on your behalf during the last 10 minutes or so of the lecture. And with that, Amanda, thank you so much. We really appreciate you being here, and we're really looking forward to your presentation.
Great, thank you. So as Jack said, I'm talking today about the challenges and opportunities of incorporating diverse populations in multi-omics research. This wasn't the original topic I planned when I first agreed to present this series, but I thought given the environment we're working in right now, that this is a very important topic and something I wanted to really discuss. And it's a topic that I find very important in my research. I didn't make it as high level as I think some of the previous talks in this series has been, as molecular as some of the previous talks have been, because I wanted to make sure this remained accessible and understandable to a wider population. So hopefully you can join me as we learn a little bit about it and think more critically about it. So what is diversity? That's a word that's used a lot right now. And what it really means is that it's any dimension that can be used to differentiate groups from people or one another. So when we say diversity, usually we think race, ethnicity. And as far as multi-omics research goes, this is a very important aspect of diversity. Racial components represent a social marker of genetic ancestry, and genetic ancestry is one of the only true confounders of genomics research. So it is a very important piece. But especially when we broaden into the broader multi-omics sphere, there's a lot more to diverse research than just race ethnicity. And so three of the other kind of big hitters are age, gender or sex, and socioeconomic status. Those ones are frequently used, well understood, but it really goes even beyond this. So if you think about all the different things that can differentiate populations that could be important for molecular or clinical research, you can think about things like disease stage. You know, are we talking about advanced cancers, early cancers, pre-diabetes, advanced diabetes, social cultural conditions? Diet plays a huge impact in metabolomics research. What kind of diet does this population eat versus that population? If you're looking at the downstream omics, so we have, you know, genomics at the kind of top of the line, but you got epigenomics, transcriptomics, proteomics, metabolomics, and it keeps going. Genetics itself can be a diversity factor. You know, we want to make sure that not everyone has the same genotype, or you're not going to see the same differentiations. Rural, urban, or geographic location, you can have access to services, pollution factors. Those can all influence omics research. Comorbid conditions, you know, think about whether or not you want somebody who's diabetic. I have obese and underweight listed here, but those also kind of go into comorbid conditions. You can look at disabilities or sexual orientation or behavioral norms, thinking about who's participating in your research and why. You know, are they more educated, more health literate? Do they have better access to services? Is somebody who participates in research more likely to be adherent to medication? Things like that. So all of these pieces form the really complex topic of diversity in research. So it's a lot more than just, are we looking at somebody who's European American, African American? It's more than that, and it's important to consider that aspect of it. And why do we care? So why is diversity important when we're doing multi-omics research? So first of all, better representation. Then you have more powerful studies, decreased health disparities, a better understanding of disease etiology or biology, and a wider application. I'm going to touch on each of these in a little bit more depth here. So starting with better representation. And so here I have a graph that is been published in Nature Medicine. And this is the makeup, the race ethnic makeup of the all the populations in the GWAS catalog up to 2021. So it's not currently up to date, but it is very recent and pretty accurate. And you can see trends here. And if you look really closely, you can see we have grown our non-European sample substantially in the last 10 years or so. That's the blue colors and below, basically, in the graph. They represent a much larger proportion than they did, say, 20 years ago. But it's still a very, very small piece of the overall pie. And when you look at the global population, it's not representative. Most of our molecular data is based on European samples. And Europeans make up a very small piece of the global pie. And our number is getting even bigger here. And you think about why that is. We've had access to things like the UK Biobank or some of these other really, really large biobank studies. A lot of them are predominantly white. And because of this, you see this exponential growth in European descent populations and a very marginal growth comparatively in these more diverse populations. And so if we make a better effort to include the studies that are down here on this little bottom corner of the graph, then we have a lot better chance of representing what's truly going on the global population. And this is an example in race ethnicity, as we've talked about. It's kind of the easy hitter when it comes to diversity, but it applies outside of that too. You know, if you're studying a population that only eats the Mediterranean diet or was applied to the salt diet, then you're going to have a really hard time generalizing those results back to, say, the broader US population that eats very differently than that. And this isn't particularly important in that case in metabolomics, which are heavily influenced by diet. So really, you got to think outside of race ethnicity. You want to represent the group that you want to apply it to. And that's hard to do if you're only focused on one group. The other piece is more powerful studies. So this is just a really rudimentary linkage disequilibrium map that was published in the JCI, and I like this figure even though I didn't make it, so I gladly use it and cite them. And basically, it's just showing if you're studying Europeans and then you're studying Africans, Europeans have much larger linkage disequilibrium patterns here. So they have much larger sections of the genome that are inherited together than Africans, who represent a much longer, basically ancestral tree. And so if you're looking, this blue star here, this is what we would find in a genetic study. This is the SNP that we would find as associated with whatever disease we're looking at. But it's not the SNP that actually causes an effect. Most likely, it doesn't do anything. But it tags the SNP that does. If you study it in Europeans, all we know is that SNP is somewhere in this big range. If you study it in Africans, you're not even going to find this one, because this SNP isn't linked to this one. Instead, you're going to find this guy. So if you're studying it in Africans, you know that it's in this little region somewhere. But if you look in both populations together, so if you've studied it in Europeans and in Africans, if you have a diverse set, then you can narrow it even further and say, okay, got to be somewhere in here. The smaller region, you can eliminate a little bit on this end, you can eliminate a lot on that end, and you know your causal SNP is somewhere in here. And so you've increased the power of your study to truly detect the right SNP by just looking in two different populations. And this is really, really kind of a critical piece in how omics research is done. The other aspect that it's really important to consider is health disparities when it comes to using diversity in molecular research. And really what it is we have a toggle switch in our hands when we're doing multi-omics research. If we continue to study just European populations, we're going to actually increase health disparities. So we're gonna create knowledge that can lead to treatments, medicines, prevention, and we're going to create all that knowledge in a group that already has advantages in medical research. If we start including more diverse populations, we can actually toggle the switch down so that we start to decrease areas of health disparity. And this is an example that I really like from genetics research, which is the APOL1 SNP in chronic kidney disease. African Americans suffer higher rates of chronic kidney disease than European Americans, and we didn't understand why for a very long time. Genetics research comes to the table and starts looking at African Americans specifically and finds these APOL1 risk variants. And there's generally two variants that are of particular concern in APOL1. And they explain a lot of the difference in why African Americans are at much higher risk. But if you look at this graph, which shows the frequency of these variants in different populations, you can understand why we really didn't see it. We had no idea APOL1 was really that important, because looking in Europeans, they're all less than 1%. Very few Europeans have the APOL1 risk variants. But if you start looking at African descent populations, so if you look in the Caribbean, if you look in Northern Central America, if you look in African Americans, and then if you look in populations in Africa where these American-based African descent populations tended to stem from, so West Africa, Nigeria, you start to see much higher rates of APOL1. And we wouldn't know this, we wouldn't understand this, we wouldn't be able to screen African Americans for APOL1 if we hadn't studied them. So you really have this opportunity to decrease health disparities if you look in the right populations. The other piece, and this kind of correlates a little bit with the previous as well, is understanding the etiology of disease and human biology. So the same way we now understand APOL1 is involved in chronic kidney disease, we also have an example here with cholesterol. And so similarly, we were studying cholesterol in whites and not seeing what we were looking for. We start studying African Americans and we discovered the PCSK9 gene. PCSK9 is involved in the metabolism of LDL. Without studying African Americans, we never find this gene. Because we've studied this gene, we now understand more about the metabolism pathway of LDL, and we've been able to take that and apply it in clinic, and we now have PCSK9 inhibitors, which have made a significant difference in cholesterol management. And so this is another example of why we really do need to study diverse populations. And then this last one here is the wider application. So evidence-based medicine is only as effective as the evidence that it relies on. This is very clear that you can't apply something to a population if you don't know anything about that population. So the more diverse the evidence base, the broader the application can be. So if you think about it, you can apply things to global populations, you can apply to different specialty populations. In particular, I'm thinking pregnant women or children. They have different omics measures, different way, their molecular system is different, even if their genetics is more inherently the same. And so this really comes into play with multi-omics when we think about some of these more layers to the population, obese populations, people who eat different diets, who live in different parts of the country that face different pollution exposures. All of these things change how you can apply your research and try and get it translated into clinical or public health practice. And frankly, there's a monetary component to this, and we live in a world where money matters. If you can argue to the pharmaceutical industry, the funding industry, that you can make more money if you study better populations, then you've got a good argument, and it's not a wrong one. And so that's something to really consider when you're choosing the populations to study. So this is one of my favorite case studies. This is clopidogrel or Plavix. And this is a great example of why studying diverse populations makes a difference. So clopidogrel is an antiplatelet drug that's used either in the primary prevention of stroke and heart attack in people with AFib, or it's used in secondary prevention for people who have had certain heart conditions or heart procedures such as PCI or angioplasty. And it's used to lower your risk of adverse heart um, heart conditions, but it has a very narrow therapeutic window. And so if your active metabolite, so if your clopidogrel active metabolite is too high, then you are lowering your platelet reactivity by too much, and you're increasing your risk of bleeding. So basically, you're making it so that your blood can't clot. That's dangerous. On the other end, if you make your platelet reactivity too high, if you have too little of the active metabolite, then you increase your risk of major adverse cardiac events. Also a major problem. And what it turns out is genetic plays a major role in whether or not the standard dose of clopidogrel can get you into this narrow therapeutic window. But if we hadn't studied diverse populations, we wouldn't understand why genetics plays a critical role. So it plays right into our story that we're telling here. So here is basically the molecular pathway that clopidogrel follows, from ingestion in the intestine, moves over to the liver where it's metabolized by multiple CYP genes. Then it moves on to become the active metabolite here, along with a few inactive metabolites, where it then binds to the P2Y12 receptor and leads to platelet activation. What we're interested in is this CYP2C19 gene. So CYP2C19 plays a very important role in the metabolism of clopidogrel in the liver. But it, like many of the CYP genes, has many variants, and they play an important role in how well you metabolize clopidogrel. So the Clinical Pharmacogenetics Consortium or Implementation Consortium, or CPIC, has categorized all of these CYP2C19 variants known of to date into these multiple categories based on their predicted function. So you have anything from ultra rapid metabolizers all the way down to poor metabolizers. And this is what we're interested in, are these poor metabolizers. So these CYP2C19 diplotypes, these are the alleles that are most commonly associated with these phenotypes. It's not an extensive list. There are some very, very rare ones that I didn't add to the list, but overall, poor metabolizers mean you either have 2 copies of STAR2, 2 copies of STAR3, or a STAR2 and a STAR3. So why are we worried about that? As you can see here, most people across most populations are normal metabolizers. So it's not something that when we brought clopidogrel to the market, we understood right away, because we were seeing the vast majority of people are in the therapeutic window, are doing what we expect. This population, this is oceanic individuals. They are different than everyone else when it comes to CYP2C19 frequencies. What we're interested in, though, is this poor metabolizer group. So if we pull that out here, only 2% of European descent populations are poor metabolizers. So if we're just studying whites, we're not finding out why CYP2C19 is important. We're not going to understand it. For every 100 people we study, 2 of them on average are going to be a poor metabolizer. And it's going to take us a very large sample to get enough of them to really see, oh, hey, it was genetic. That's why. It's not one of the other million differences between these people. However, if we look at some of these other populations, then this poor metabolizer group becomes much more critical to understand. So we'd already talked about how the oceanic population differs from most of the rest of the world, where 57% of them are actually going to be poor metabolizers of clopidogrel. So this is maybe something that maybe not the drug that we want to export to countries in Oceania. The other group is East Asian. So this is a very large population. 13% of East Asians on average are not going to respond to clopidogrel. And they're going to be at increased risk of adverse cardiac events. So what does this ultimately mean for research, though? Well, to discover the subset of people who don't respond well to clopidogrel, if you're studying European Americans, you have to study 7 times as many European Americans as East Asians in order to actually get a population size big enough to adequately identify the star 2 and star 3 alleles in this population. You need 28 times more Europeans than Oceania. So if you can somehow come up with a population of, people from Micronesia, Fiji, the island nations of Oceania, then you have a huge advantage and need a much smaller population because these star 2 and star 3 alleles are so much more common in those populations. So let's put some actual numbers behind this. I've done some simulation work that shows for a lot of pharmacogenomic studies like this, you need populations of approximately 150,000 people to identify at a sufficient significance level a SNP that is approximately a minor allele frequency of 25%. If you have a diplotype of 2%, you're talking less than 1% minor allele frequencies. You're going to need a populations that are 20 times 150,000. You're going to need populations well over 1 million to identify this SNP in Europeans. If you're talking East Asians, all of a sudden our numbers are much, much smaller, but still big. For East Asians, where we did actually find this SNP, we still needed to really either pick and choose. We need to do some kind of case control where we're really picking out the poor metabolizers of clopidogrel from a clinical sample to really isolate the group. Or if you're studying it on a general population size, you need, 700,000. If you're talking oceanic populations where it's present in 57% of individuals, all of a sudden now we're talking minor allele frequencies that are excellent, and you could easily get away with 100,000, much smaller sample. So if you have limited resources, you're going to see much better return for your dollar if you can study diverse populations where you can get this kind of power exchange. And so this is just a really good, I think, example of where diversity makes a measurable impact in how we identify things. But, we're talking about multi-omics, and we've talked about how diversity is so much more than just race, ethnicity, or genetic ancestry. So what about these other omics measures or other forms of diversity? So I've picked out just a few examples of where these also can be seen. These differences are important. So we started with genetics, and if we're thinking kind of about the omics pathway, our next step is really epigenetics. And so methylation patterns, we already know that they can vary by race, ethnicity, because we've definitely seen evidence of that. But now here we see that they also vary by age. So when you're studying methylation patterns, it's important to consider the age, not just the genetic ancestry of your population. So here they're looking at a group of nonagenarians or people over 90 compared to younger controls. And they've looked at 8 different CpG methylation sites. And you can see significant differences in the level of methylation between our extreme elderly population and our younger controls. And so we see age matters when you're looking at methylation patterns. And so you can then assume, because methylation patterns affect transcriptomics affect proteomics affects metabolomics, you can assume age is going to affect everything downstream as well. Looking at our next kind of step down, transcriptomics, here's an example where gender or sex is affecting your transcriptome patterns. And so here we have the logarithm of the fold change for men, which are the blue, or women, which are the orange, in different transcripts, or I believe these are RNAs, in multiple sclerosis. And so you can see that both in the blood and the brain, which is where they took their two samples from, they can find examples where women and men have different changes in the transcript patterns between people with and without multiple sclerosis is what this is. So here, sex is playing an impact on our transcript patterns, which we can then assume is also going to impact our proteome patterns, which will also impact our metabolomics patterns, and so on and so forth. Moving down the line, I looked at proteomics to find a good example of how another form of diversity in research can affect it. And this time I went with disease stage. So here you can see the number of proteins that are differentially expressed, either over or under, in people with high grade or high stage cancers compared to their counterparts with lower stage cancers. So for example, if you look here at a high grade lung cancer, 824 of the proteins that they studied were overexpressed in the higher grade cancer, and 919 were underexpressed in the high grade cancers compared to a lower grade cancer. So you want to think about, when you're studying proteomics, metabolomics, anything on this pathway, what disease stage are we at? What variation of the disease are we at? We have a lot of diseases that have multiple etiologies, and the different etiologies, we can expect different molecular signatures. That's very common, a very common expectation in the field. And so you really want to think about how heterogeneous is your disease exposure. And then my final example that I got brought out was in metabolomics. So I'm continuing to move down the pathway. And this one, I wanted to look at BMI. So one of our comorbid conditions that you can think about. And here you can see this is kind of hard to see because I know it's small, but here you have a BMI under 30. So this study was looking at overweight and obese. So you have people whose BMI is in the overweight category. And then here, there we go. Here you have people whose BMI is greater than 40. So you have the morbidly obese group. And then in between you have obese individuals. And you can see that they looked at different metabolites. Here we show 16 different metabolites. And they found linear relationships. So there's kind of a dose-response relationship between these metabolite levels and your BMI. And some of them go up and some of them go down. But BMI is going to make a difference in your measurement of your metabolite. So it's something that you need to think about when designing a study of metabolites. Is everyone overweight? Is everyone underweight? How generalizable is this finding going to be after you produce your results? So if we understand why diversity is so important, we also need to think about, why aren't we just including it all the time? So what are the barriers to having a diverse study? And when I'm designing my studies, I like to think about it in two buckets. So I think about the barriers that are in my research design bucket and the barriers that are in my social cultural bucket. So thinking research design. First, there's a lack of appropriate reference populations, particularly in multi-omics research. We rely heavily on replication. So we study a lot of things that have a lot of tests. We have a massive multiple testing burden in multi-omics research. And so when you are doing that, there's a higher chance for false positives than when you're studying a single individual outcome. Because of that, we like to replicate. If we see it twice, it's more likely to be true. But how do you replicate something if you can't find a similar population? So, when we're thinking about it from the race ethnic point, which is our easiest diversity group to kind of think through, we saw that graph earlier where you see a massive amount of Europeans, but much smaller portions of these other groups. And so if you're studying, say, African Americans, you might need every single African American you can get your hand on for the discovery sample. So how do you replicate it? Because there's no one else left for you to replicate it in that's the same. You can try and replicate it in Europeans or in Africans, but Africans aren't the same as African Americans. Europeans aren't the same as African Americans. So if it doesn't replicate, does that mean it's not true? Or does that just mean that it's population specific, or you're not seeing the right linkage disequilibrium patterns? And so without the right reference population, it's a lot harder to replicate. The other piece of that is imputation, which is so critical in multi-omics research. If you're trying to impute genetic data, transcriptomics data, metabolite data, whatever type of omics you want to impute, you need the right reference group. So if you're trying to impute African American genetics and you're using Europeans to do it, you're not going to get the right genetic pattern. The same goes for transcriptomics, metabolomics. We can impute at every stage of the omics pathway, some with better precision than others. But if you're trying to do it, say you're trying to impute a metabolite that is heavily influenced by diet, and you're imputing it to someone who, eats a very different diet, is a solid meat eater, and you're trying to study it in vegetarians. Like that, something like that would also be a major problem with your imputation pattern. And this can come in other forms. One of the big ones imputing transcriptomics measures is tissues. We expect different transcript profiles in different tissues of the body. Most of our reference panel comes from blood tissue. Obviously, that's the easiest to get. There's not a lot of people who want to give you a live sample of their heart or their brain. And so when we're imputing, we can't assume that that works for a brain phenotype. So like I said, this diversity really affects multi-omics research when it comes to reference populations. The other aspect, and this kind of ties in a little bit, is this small sample size. Multi-omics research requires larger samples than most other epidemiologic studies. That's because we're studying so many things at once, so we need so many different, so many more people to kind of overcome that multiple testing burden, overcome the false positives, and get enough people that we can actually run a statistical model on it. Because we need that many people, it's harder to study these smaller and smaller groups. So even though we're now into the millions, and we have millions of people with genetics data, for example, if you're trying to break these down more and more and more, you can break it down to East Asians versus Europeans and still have millions. But what if you want to study Chinese versus Koreans versus Japanese. Like not all East Asians are the same. Not all Europeans are the same. And the more you try and break it down, the harder and harder it gets to have a sufficient sample size. This is even more prevalent when it comes to non-genetic omics measures. So genetics is becoming cheaper and easier to get. So we have it on a whole lot of populations. But what about transcriptomics? It still costs a lot to run RNA-seq. Metabolomics profiles still cost quite a bit of money to generate. So these downstream omics measures are present in even fewer individuals than our genetics measures. Then another thing that we think about is what public data resources are available. And these tie in to the above points as well. And it's critical. We really rely on public data for multiple stages of omics research. First, just the data sets. And this is something that's really important to understand is that, these publicly available data sets that have been so wonderfully maintained are critical to being able to produce these large populations because we can combine so many more data points, so many more individuals if we can combine these big public data sets. And so that's important. There's also the piece in omics research about these publicly curated resources. I'm thinking things like ENCODE or Brainiac or the UCSC genome browser, things like that, where we can go if we have limited resources. Say we find something in a genetic study, but we don't have the money or the tissue to run a transcriptomic analysis on this data. We can go into these other resources that are publicly available and do some bioinformatic mining, figure out what the pathway is that we're interested in based on our findings. And it's a wonderful resource in omics research because it is still so hard to get these downstream omics measures a lot of the time. But it goes back to the whole issue of appropriate reference populations. If our ENCODE data, for example, is on the wrong tissue or the wrong race ethnicity or from someone who only eats spaghetti for dinner, then we have less generalizability and our results aren't what we actually think they are. And so it's really important to kind of know what's available and try and enhance these public data sets. And then the last piece is that epidemiologic research values homogeneity. The more similar your study, the better your validity and the better your statistical power when you analyze the data. This is the reason that a lot of pharmaceutical companies will have really strict inclusion and exclusion criteria on their clinical research when they're submitting applications to the FDA. This is the reason that a lot of studies have really clear, population that they're trying to access. This is the reason why we recruit similar people to different studies. Homogeneity is power in epidemiologic research. Diversity is inherently heterogeneous. So we have a little bit of a balance here. If you study it in multiple populations, we've already talked about how that can increase your power, which is great. But it also can hinder your power because if your population is too different, then you're not going to see the same effects at the same rate. And so one way we deal with this in omics research is to study it both together and separate. So like we'll run race specific analyses, and then we'll run a transethnic analysis. And that way we can really balance the larger population in the transethnic analysis and get the power hit from the larger population, but also get the power hits from separating out each individual ethnicity. So we have more homogeneous populations, even if they're smaller. Okay, so then my other bucket that I talked about is these social cultural concerns. And so the big one that we encounter a lot, especially in molecular research, is mistrust. There's a lot of mistrust in clinical research among underserved populations. Two big examples of that are going to be your Native American and African American communities. They've been exploited by research in the past. They've been exploited by genetics research in the past. And so that makes them really hesitant to participate in clinical studies. It's also common to see mistrust among highly publicized or highly stigmatized research. So a couple examples of this from recently, mRNA vaccines are a big one. So mRNA is all over the news. You go to somebody and say, hey, I want to run a transcriptomic analysis. I want to recruit you into to study your RNA. They immediately step back because what they know about RNA is linked to that news story they saw about mRNA vaccines. Not necessarily the same thing, but they link it in their head. And so there's this issue of overcoming this highly publicized topic. Another example is mental health research. We know there's a stigma around mental health treatment in this country, in a lot of the world. So if you're trying to study mental health and trying to recruit somebody into a study to look at, say, the genetics of schizophrenia, people with schizophrenia are going to be more hesitant to participate. People without are going to be hesitant to participate. What if somebody thinks I have it? What if somebody discovers I have it? What if the data isn't as secure as you think it is? What if we find out something about it I don't want to know? So these heavily stigmatized areas also create a cloud of mistrust. Another thing to think about is that population face time and resource constraints. So thinking about having time for clinic visits, having time for treatment. Poor and rural populations in particular face these constraints, and more rural populations in different parts of the globe. So it goes both ways. The individuals don't necessarily have time or resources to come to us. We might not have the time or resources to get all the way to them. So that resource constraint can go both ways. Cultural beliefs or lack of scientific literacy. The scientific literacy goes a little bit back to that stigmatized issue too. If they don't understand it, they might link it to something that's not quite the same in their head. You might have to really educate a population. Another thing is cultural beliefs. A good example of this in omics research is the Hispanic community. Hispanic individuals tend to have a very strong sense of their history, where they come from, what the stories of their people are. And they don't want you to come in, collect their genetic data and tell them, oh, wait, we ran a genetic ancestry analysis and we say that you're actually European when culturally you believe that you come from a Native American community. They don't want that to happen. They're very strong in their beliefs of where they come from. And so when we work with Hispanic communities to collect genetic data, we learn about that. We talk to them about that, and we go, okay, we will make a commitment not to run those ancestral analyses. We won't publish anything that contradicts your belief in yourself. That's not what this is about. We want to run the research that you approve of using your information for. But that's something that you have to overcome. And then the last one is a reality. It's particularly noticeable today in the current kind of political climate we're in right now, but it also goes way beyond just the current US political climate. So the reality is that a significant portion of global research dollars comes from government resources. And so it's hard to separate political agendas from scientific agendas. And we have that example in the US right now where it's challenging, but there's other examples that are like Brexit's a good one. You had a lot of scientists in the UK who were suddenly pulled from their resources, from their monetary support, because they were getting it from the EU. UK pulls out of the EU, all of a sudden they lose their resources. That had nothing to do with the science, but it was a political reality. And things like that happen. And expanding diversity requires money and resources. And so the reality is that we have to try and balance political and financial climates with our research in order to fully expand diversity. And so I don't want to leave on a negative tone. So I want to talk about ways we can overcome this, because we've already talked about why it's important that we do. One big way is to continue to just grow the size of our studies. Make more use of biobanks, create biobanks in more populations that are more diverse. You can use adaptive study design, where you start with a population, and as you grow and learn, you grow into new populations. So you start recruiting from different groups as you learn more about what you need. Consider really carefully when you're designing your study, what diversity is important. Is this something where race ethnicity is important? Are you looking at a metabolomic study where diet does really heavily influence your outcome? Are you studying something where diabetes, for example, is heavily influential in both your omic and your outcome? So maybe you want to make sure you have a sufficient amount of diabetics, non-diabetics, pre-diabetics, things like that. So it's really important when you're designing your study and figuring out what population you want, that you think about where diversity can help. And this is another great one. It's not something that most of us as molecular researchers are good at, but it's something that we can partner with people to really get at, which is using qualitative research to identify why certain populations are difficult to include. So what specific barriers do you need to overcome? For example, we talked about the Hispanic community. And so by talking with them, by using that qualitative piece, we were able to learn about their cultural history and their belief in their own etiology. And so we could figure out, okay, what do we need to do to get them involved? And figure out a way to say, we don't want to study your ancestry, we want to study your disease state, things like that. And that ties very closely into then building relationships with the community. So if you want to expand into new communities, if you want to expand into more diverse communities, you want to make sure you have that relationship. Participants aren't going to trust a study that they don't have a relationship with. So you need to build that. You need to engage diverse physicians, engage diverse communities, build community activities. A great example of that is working with Native American populations. They're very hesitant to work with federally funded research because of some really questionable decisions made in the past by researchers and federal authorities. And so because of that, to work with Native American communities, a lot of times you have to sit down with the elders of the community and talk about what the data will be used for, how it will be used, what they're okay with, what they're not okay with, and build your study around that. And then finally, go global. We live in a global society. We live in a global community. So our research can affect everyone on the globe. So why aren't we researching all over? We have great data in a lot of countries, but for example, most of Africa is still poorly covered by research studies, specifically molecular ones. And so don't just choose your study site for ease or what was population, what economic reasons. For example, you don't have to spend any money to get this data because it's already at your university. That's great. And I'm guilty of that too, because sometimes you don't have the money to spend. But also think about it from what population's available. Like, do you need to go and get the right data to answer your question? And so that's another big piece to think about. And so what the statement I kind of want to leave you with is that diversity in healthcare research is a prerequisite to equity in health. So you're never going to achieve equity in healthcare, in public health, in any stage of the health process, unless you start early. It has to be part of your research in order to affect your outcome. And so that's all I have. And we can have a little discussion now. I think
Amanda,
I hit perfect timing.
perfect timing. Thank you, Amanda. That last thing you touched on was something that, concerning going global, was something that was just kind of had me thinking about this almost your entire talk. Is like, what has stopped the NIH, the World Health Organization, the United Nations, and basically all of their international opposite numbers to just make a decision to just start gathering genomic, transcriptomic, and basically just, having people spit into cups and mail it someplace, and it could be someplace that the world agrees upon is, politically neutral. Mail them to Geneva, and get basically the world population, these little isolated islands that we tend to focus on, and then a lot of them were in Europe. Is I think it's doing us a disservice. What if we just said the population is the planet, is the 10 billion people that live on the planet Earth, and that's our population, not people who are from, Finland, or people who are from, Ghana or from
Mhm.
Ireland. It's basically the planet. If we just started doing that and we just made it a global resource, we'd always have the population at our fingertips, the population writ large with a capital P, instead of these, isolated things. What's stopping that from happening?
I mean, there has been efforts to do that. There's efforts to collect, gosh, now I'm blanking on the name, but there's a massive undertaking to get genetic data out of Africa. But for one, where does all the funding come from? Um, how do you access the populations? A lot of these populations, they can't just spit in a cup and mail it. You have to physically go to them, get them to spit in the cup, collect all the data. How do you do a really efficient, informed consent if they don't have the scientific literacy to understand what you're asking them? Um, what if you don't understand their cultural norms? There are populations in Central America and South America that we've looked to study that don't really believe in science. And so then trying to do a scientific study on them is really hard because we don't understand how or why to do it. And so I think a lot of it comes down to the massive transdisciplinary effort it needs to be. You need the funding source, you need the scientists, but you need a variety of scientists. You need the molecular biologists, you need the anthropologists, you need the the sociologists. You need a really big undertaking to really do this on the right scale. And I think it's just at this point it's a resource limit, um, unfortunately.
Let's say that something like that were to happen. Let's say that, certain billionaires who have nothing better to do actually put their money towards something for the
Mhm.
world good and paid for this. And you had all the data from every country, from, the entire population. What sort of data challenges would that present in your view?
Oh gosh, now we're getting into the whole big data issue. Um, you know, where do you store it? How do you store it? Um, how do you analyze it? The Google Cloud, Amazon Cloud, they're wonderful resources, but are they big enough for a data set that is that big or that diverse? Um, that's another thing that you really need to think about when you're building out that large of it and that diverse of a data set is where do we analyze it and how do we store it?
Absolutely. It's going to be the biggest of big data. But, maybe
Hmm.
if we got started today, we might solve some of those problems. And that might be kind of interesting.
Would be wonderful.
Well, Amanda Seyerle from the University of North Carolina, thank you so much for presenting your work and your thoughts, and some of these, very thought-provoking things for us to kind of chew on. I really appreciate it. Thanks for joining us. Everyone have a wonderful weekend, and don't forget those Biomedical Data Science Innovation Lab application materials are still being welcomed up until Valentine's Day. So if you're interested, think you are a junior faculty working at the intersection of kind of a multi-omics and data science, or either one or the other, please send in your materials. We'd be delighted to take a look, and would be very interested in considering having you. So have a wonderful weekend.